NCERT Biology textbook for 12th standard, chapter number 5th, the principles of inheritance and the variation. Have you ever wondered why an elephant always gives the birth only to the baby of elephant and not some other animals? Or why a mango seed from only a mango plant and not any other plant? Give that they do, are the offsprings identical to their parents, or do they some shows differences in the sum of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different? These and the several related questions are dealt with scientifically in a branch of the biology known as the genetics. This subject deals with the inheritance as well as the variation of the characters from parent to the offsprings. Inheritance is a process by which the characters are passes on from the parents to the progeny. It is the basis of heredity. Variation is the degree by which the progeny differ from their parents. Humans knew from as early as the 8000 to the 1000 BC that one of the causes of the variation was hidden in a sexual reproduction. They exploited the variations that were naturally present in the wild population of the plant and animals to selectively breed and select for the organisms that possess the desirable characters. For example, through the artificial selection and the domestication from the ancestral wild cows, we have well-known Indian breeders, for example the Sahiwal cow in Punjab, and we must however recognize that though our ancestors knew about the inheritance of the characters and the variations, they had a very little idea about the scientific basis of their phenomena. 5.1 is a Mendel's law of inheritance. It was during the mid-19th century that headway was made in the understanding of the inheritance. Gregor Mendel conducted a hybridization experiment on the garden pea for the 7 years, that is from 1856 to the 1863, and proposed the law of inheritance in living organisms. During Mendel's investigation into the inheritance pattern, it was for the first time that statistically analysis and the mathematical logic were applied to the problems in the biology. His experiment had a large sampling size which gives greater credibility to the data that he collected. Also, a confirmation of his inference from the experiment on the successive generation of his test plant proved that his result pointed to the general rule of the inheritance rather than being a unsubstantiated ideas. The Mendel investigated characters in the garden pea plants that were manifested as two opposing traits, for example the tall or a dwarf plant, yellow or a green seeds. This allows him to set up the basic framework of the rules governing the inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. The Mendel conducts such artificial pollination or a cross-pollination experiments using several true breeding pea lines. A true breeding line is one that having undergone the continuous self-pollination and shows the stable trait inheritance and the compression for the several generations. The Mendel secreted the 14 true breeding pea plant varieties as pairs which were similar except for one character with a contrasting trait. Some of the contrasting traits selected were the smooth or a wrinkled seeds, yellow or a green seeds, inflated means the full or a constricted green or yellow pods, and a tall or a dwarf plants. The 5.2 is the inheritance of the one gene. The contrasting trait studied by the Mendel in pea is a character. First character is the stem height, it may be tall or dwarf. The second is the flower color is violet or white. The third is the flower position, the axile or a terminal. The fourth character is a pod shape, is inflated or constricted. The fifth character is a pod color, is a green or yellow. The sixth character is a seed shape, is a round or wrinkle. The seventh last one character is the seed color, that is the yellow or green. Now the 5.2 is the inheritance of the one gene. Let us take the example of the one such hybridization experiment carried out by the Mendel, where he crossed the tall and the dwarf pea plant to study the inheritance of the one gene. He collected the seeds produced as a result of his cross and grew them to generate the plants of the first hybrid generation, and this generation is also called as a filial progeny or the F1. The Mendel observed that all the F1 progeny plants were tall and like one of its parent, none were the dwarf. He made similar observations of the other pair of the trait. He found that the F1 always resembled either one of the parent and that the trait of the other plant was not seen in them. The Mendel then self-pollinated the tall F1 plants. To his surprise, found that in the filial second generation, some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of the plant that were the dwarf were the 1/4 of the F2 plants, while the 3/4 of the F2 plants were tall. The tall and the dwarf traits were identical to their parental types and did not show any blending, that is, all offsprings were either tall or a dwarf. None were in between height. Similar results were obtained with the other traits that he studied. The only one of the parental trait was expressed in the F1 generations, while at the F2 stage both the traits were expressed in the proportion the 3:1. The contrasting trait did not show any blending at either F1 or F0 stage. Based on these observations, the Mendel proposed that something was being stably passed down, the unchanged from the parents to the offspring through the gametes and over a successive generations. He called these things as a factors, and now we call them as a gene. The gene therefore are the unit of the inheritance, and they contains the information that is required to express a particular trait in an organism. The gene that codes for the pair of the contrasting trait are known as the allele. That is, they are the slightly different forms of the same gene. If we use the alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabets of the other traits. For example, in case of the characters of the height, the T is used for the tall trait and the small t for the dwarf, and the capital T and the small t are allele of each other. Hence, in plants, the pair of the allele for heights would be the capital T, capital T small t, or a small t, small t. The Mendel also proposed that in a true breeding, the tall or a dwarf pea varieties are allelic pair of the genes for the height are the identical or homozygous, that is the capital T and the small t respectively. The capital T and the small t are called as a genotype of the plant, while the descriptive term is a tall and the dwarf are the phenotype. What then would be the phenotype of the plant that had a genotype capital T small t? As Mendel found the phenotype of the F1 heterozygotes, the capital T small t, to be exactly like to the capital T parents in the appearance, and he proposed that in pair of the dissimilar factors, the one dominant and the other as in the F1, and hence is called as a dominant factors, while the other factor is a recessive. In this case, the capital T for the tallness is a dominant over the small t for the dwarfness, that is recessive. He observed the identical behavior for all the other characters or a trait pair that he studied. It is convenient and logical to use the capital and lower case of and the alphabetical symbols To remember this concept of the dominance and the recessiveness, do not use the capital T for the tall and the small d for the dwarf, because we'll find it difficult to remember whether the capital T and the D are the allele of the same genes or a characters or not. The allele can be similar as in the case of the homozygote, that it is the capital T and the small t, or can be dissimilar as in the case of the heterozygotes, that is the capital T small t. Since the capital T small t plant is the heterozygous for the gene controlling the one characters, that is the height, it is the monohybrid, and the cross between the capital T and the small t is a monohybrid cross. From the observation that recessive parental trait is expressed without any blending in the F2 generation, we can infer that when a tall and a dwarf plant produces the gametes by the process of the meiosis, and the allele of the parental pair separate or segregate from each other, and only one allele is transmitted to the gametes. This segregation of the allele is a random process, and so there is a 50% chances of the gamete containing the either allele as has been verified by the result of the crossing. In this way, the gametes of the tall capital T plant have the allele of the capital T, and the gametes of the dwarf, that is the small t plant, have the allele of the small t. During the fertilization, the two alleles, the capital T from the one parent says through the pollen, and the small t from the other parents then through the eggs, are united to produce the zygote that have the one capital T allele and the one small t allele. In other words, the hybrid have the capital T small t, since these hybrids contain the alleles which express the contrasting traits. The plant are the heterozygous. The production of the gametes by the parents and forms them, and the formation of the zygote, the F1 and the F2 plants, can be understood from the diagram called as a Punnett square, as shown in figure. It was developed by the British geneticists Reginald C. Punnett. It is the graphical representation to calculate the probability of all possible genotype of the offspring in the genetic cross. The possible gametes are written on two sides, usually the top row and the left columns. All possesses or possible combinations are represented in a box below in the square, which generates the square output form. The Punnett square shown that potential parental tall, that is the capital T male, and the dwarf is a small t is a female plant. The gamete produced by them and the F1, the capital T small t progeny. The F1 plants of the genotype, that is the capital T small t, are the self-pollinated. The symbols are used to denote the female egg and the male pollen of the F1 generations respectively. The F1 plant, the genotype capital T small t, when self-pollinated, produces the gamete of the genotype of the capital T and the small t in equal proportions. And when the fertilization takes place, the pollen grain of the genotype of the capital T that has a 50% chances to pollinate the egg of the genotype capital T as well as the genotype small t. Also, the pollen grain of the genotype small t have the 50% chance of the pollinating egg of the genotype capital T as well as of the genotype small t. As a result, the random fertilization, the resultant zygote can be of the genotype, that is the capital T, small t, or the small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization leads to the capital T and 1/2 leads to the capital T small t and 1/4 to the small t. Although the F1 have the genotype of the capital T small t, but the phenotypic character seen is a tall. All F2, 3/4 of the plants are the tall, where the sum of them are the capital T, while the other are the capital T small t. Externally, it is not possible to distinguish between the plants with the genotype of the capital T and the capital T small t. Hence, within the genotypic pair, the capital T small t, only one character, that is the capital T tall, is expressed. Hence, the character, that is the capital T or a tall, is said to be the dominant over the other allele, that is the small t or a dwarf character. It is thus due to this dominance of the one character over the other that all F1 are the tall and thought the genotype, and though the genotype is the capital T small t. And in the F2, that is the 3/4 of the plants are the tall, and through the genotypically 1/2 are the capital T small t and only 1/4 are the capital T. This leads to the phenotypic ratio, that is the 3/4 of the tall as to the 1/4 capital T plus 1/2 capital T small t, and the 1/4 are the small t. That is the 3:1 ratio, but the genotypic ratio is the 1:2:1. The 1/4 as to 1/2 as to 1/4 ratio, that is the capital T as to capital T small t as to capital small t, is a mathematical condensable to the form of the binomial expressions, that is the ax plus by bracket square, and that has the gamete bearing the genes the capital T small t is the equal frequency of the 1/2. The expression is expanded as given below. The 1/2 capital T plus 1/2 small t bracket square is equal to 1/2 capital T plus 1/2 small t into 1/2 capital T plus 1/2 small t is equal to 1/4 capital T plus 1/2 small t capital T small t plus 1/4 of the small t. The Mendel self-pollinated the F2 plants are found that the dwarf F2 plants continue to generate dwarf plants in F3 and the F4 generation. He concluded that the genotype of the dwarf was a homozygous, that is the small t. What do you think he would have got had he self-pollinated and the tall F2 plants? From the preceding paragraphs, it is clear that though the genotypic ratios can be calculated using the mathematical probabilities by the simply knowing at the phenotype of the dominant traits, it is not possible to know the genotypic composition. That is, for example, whether a tall plant form the F1 or F2 has the capital T or a capital T small t composition cannot be predicted. Therefore, to determine the genotype of the tall plants at F2, the Mendel crosses the tall plant from the F2 with the dwarf plant, and this he called as a test cross. In typical test cross, an organism, the P plant here, showing a dominant phenotype and whose genotype is to be determined, is a cross with the recessive parents instead of the self-crossing. The progeny of such cross can be easily be analyzed to the predicts and the genotype of the test organism. The result typically test cross where violet color flowers, that is capital W, is dominant over a white color flower, that is a small w. Using the Punnett square, try to find out the nature of the offspring of the test cross and what ratio did you get. Using the genotype of this cross, can you give the genital definition for the test cross? Based on the observation on the monohybrid crosses, the Mendel proposed the two general rules of the consolidate his understanding of the inheritance in a monohybrid crosses. Today, these rules are called as a principle or a law of the inheritance. The first law or a law of dominance, and the second law or a law of the segregation. The 5.2.1 is a law of dominance. The characters are controlled by the discrete units called as factors. The factor occurs in pairs. In dissimilar pair of the factors, one member of the pair dominates, or a dominant, or other is a recessive. The law of the dominance is used to explain the expression of the only one of the parental characters in monohybrid cross in F1 and the expressions of both in the F2. It also explains the proportions of the 3:1 obtained at the F2. The 5.2.2 is a law of segregation. This law is based on the facts that allele do not show any blending and that both characters are recovered as such as the F2 generations. The one of these is not seen in the F1 stages. Though the parent contains the two alleles during the gamete formation, the factors or allele of the pair segregate from each other such that the gamete receive only one of the two factors. Of course, the homozygous parent produces all gametes that are similar, while the heterozygotes one produce the two kinds of the gametes which having the one allele with equal proportions. Now the 5.2.2.1 is incomplete dominance. When the experiments on the peas were repeated using the other traits in the other plants, it was found that sometimes the F1 had a phenotype that did not resemble that of the two parents and was in the between the two. The inheritance of the flower color is the dark flower, that is the snapdragon, and the Antirrhinum species is a good example to understand the incomplete dominance. In a cross between the true breeding red flowers, that is the capital R, and the true breeding white flower, the plant is the small r. The F1 is a capital R, small r, was a pink in color. When the F1 was self-pollinated, the F2 resulted in the following ratio is 1 is capital R is a red as to 2 capital R, small r, the pink as to 1 small r are the white. When the genotype, here the genotype ratio were the exactly as we would expect in any Mendelian monohybrid cross, but a phenotype ratio had the change from the 3:1 dominant as to recessive ratio. What happened was that the R was not completely dominant or the R, and this made it possible to distinguish the capital R, small r as a pink from the capital R that is the red, and the small r is a white. Explanation of the concept of the dominance. What exactly is the dominance? Why are the some alleles dominant and the some are the recessive? To tackle these questions, we must understand what is the gene does. Every gene, as you know by now, contains the information to express the particular traits in a diploid organism. There are the two copies of the each gene, that is as pair of the allele. Now these two alleles need not always be identical, as in the heterozygotes, one of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modified the information that a particular allele contains. Let's take an example of the gene that contains the information for the producing an enzyme. Now there are the two copies of this gene, the two allele forms. Let's us assume, as in the more common, that the normal allele produces the normal enzyme that is needed for the transformation of the substrate, that is the S, and theoretically the modified allele could be responsible for the production of the first is the normal or a less efficient enzyme, or second is a non-functional enzyme, or third is a non-enzyme at all. In the first case, the modified allele is the equivalent to the unmodified allele, that is, it will produce the same phenotype or a traits. That is a result in the transformation of the substrate, that is the capital S. Such an equivalent allele pair are very common, but if the allele produces a non-functional enzyme or a no enzyme, the phenotype may be affected. The phenotype or a traits will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to the non-functional enzyme and because no enzyme is produced. The 5.2.2.2 is a codominance. Till now we were discussing the cross where the F1 resemble there of the two parents, that is the dominance, and was in the between incomplete dominance. But in the case of the codominance, the F1 generation resemble both the parents. A good example is a different types of the red blood cells that determines the Abo blood grouping in a human being. The Abo blood groups are controlled by the gene I. The plasma membrane of the red blood cell has the sugar polymer that protrudes from the surface, and the kind of the sugar is controlled by the gene. The gene I has three alleles, the capital A, IA, IB and the small I. The allele of the capital A and capital IB produces slightly different from the sugar, while the allele of the small I does not produce any sugar. Because the humans are the diploid organisms, each person possesses any two of the three, that is the capital I gene allele, that is the capital IA and the capital IB are completely dominant over a small I. In other words, when the capital IA and the small I represents only the capital IA expresses because the capital I does not produce any sugar. And when the capital IB and the small I present, the capital IB expresses. But when the capital IA and the capital IB are present together, they both express their own types of the sugars. This is because of the codominance, and hence the red blood cells have the both capital A and capital B types of the sugar, and since there are the three different alleles and there are the 6 different codominants of these three alleles that are possible, and therefore the total of the 6 different genotypes of the human, that is the capital A, capital B, Abo blood type. How many phenotypes are possible? Now the types of the blood types of the offspring are the A, AB, B, B and O. Do you realize that the example of the Abo blood grouping also provides the good example of the multiple alleles? Here we can see that there are the more than the 2, that is the 3 alleles governing the same characters. Since in the individual the only 2 alleles can be present, the multiple allele can be found only when the population studied are made. Occasionally a single gene products may produce the more than the one effect. For example, the starch synthesis in the pea seeds is controlled by the one gene, and it has a 2 allele, that is the capital B and the small b. Starch is synthesized effectively by the capital B homozygotes, and therefore the large starch grain are produced. In contrast, the capital B small b homozygote have lesser efficiency in a starch synthesis and produces the smaller starch grains. After maturation of the seed, the capital B seed are round and the small b seed are the wrinkled. The heterozygote produce around the seed, and so the capital B seem to be dominant allele. But the starch grains produces are the intermediate size in the capital B small b seeds. So if the starch grain size is considered as a phenotype, then from the angle the alleles shows the incomplete dominance. Therefore, the dominance is not and the autonomous features of the gene or products that it has information for. It depends as much on the gene products and production of the particular phenotype from the products as it does on the particular phenotype that we choose to examine, in case more than the one phenotype is influenced by the same gene. The 5.3 is the inheritance of the 2 genes. The Mendel also worked with and the cross pea plants that differ in the two characters as seen in the cross between the pea plants that has the seeds with the yellow color and the round shape, and one that has the seed of the green color and wrinkle shape. The Mendel found that the seed resulting from the crossing of the parents that had a yellow colored and the round shaped seeds. Here, can you tell which of the character is a paired yellow or a green color and a round or a wrinkle shape was a dominant? Thus, the yellow color was a dominant over the green and the round shape dominant over a wrinkle. These results were identical to those that he got when he made the separate monohybrid crosses between the yellow and the green seeds, the plants, and between the round and the wrinkle seeded plants. Let us use the genotypic symbols that are capital Y for the dominant yellow seed color and the small y for the recessive green seed color. The capital R for the round shaped seeds and the small r for the wrinkle seed shape. The genotype of the parent can then be written as the capital R, capital Y, and the small r, capital small y. The cross between the two plants can be written down as in the figure showing the genotype of the parent plants. The gametes capital R, capital Y, and the small y until on the fertilization to produce the F1 hybrids, the capital R, capital Y, small y. When the Mendel self-hybridized the F1 plants, he found the 3/4 of the F2 plants had a yellow seeded and the 1/4 had a green. The yellow and the green color segregates in the 3:1 ratio. Round and the wrinkle seed shapes also segregate in the 3:1 ratio, just like the monohybrid cross. The 5.3.1 is a law of independent assortment. In the dihybrid cross, the phenotype round, yellow, wrinkled, yellow, round, green, and the wrinkle. The green appear in the ratio of the 9:3:3:1. Such a ratio was observed for the several pairs of the characters and the Mendel study. The ratio of the 9:3:3:1 can be derived as a combination series of the 3 yellow, 1 green, and with the 3 round and the 1 wrinkle. These derivations can be written as follows: the 3 round as to 1 wrinkle and the 3 yellow as to 1 green is equal to 9 round and the yellow as to 3 wrinkled, yellow as to 3 round, green as to 1 wrinkle, green. Based upon the such observation on the dihybrid cross, the crosses between the plant differing in the two traits, the Mendel possessed the second set of the generalization that we call the Mendel's law of independent assortment. The law states that when the two pairs of the traits are combined in a hybrid, the segregation of the one pair of the character is independent of the other pair of the character. The Punnett square can be effectively used to understand the independent segregations of the two pairs of the gene during the meiosis and the production of the egg and the pollen in the F1, that is the capital R small r and capital Y small y plants. Consider the segregation of the one pair of the gene, that is the capital R and the small r. The 50% of the gametes have the gene, that is the capital R, and the other is the 50% have capital small r. Now, beside each gametes having either the capital R small r, it should also have the allele of the capital Y small y. And important things to resemble here is that the segregation of the 50%, that is the capital R, and the 50% is the small r, is independent from the segregation of the 50% capital Y and the 50% is a small y. Therefore, the 50% of the small r bearing the gametes has the capital Y and the other is 50% has the small y. Similarly, the 50% of the capital R bearing the gametes has the capital Y and the other 50% has the small y. Thus, there are the 4 genotypes of the gametes, the 4 types of the pollen and the 4 types of the eggs. The 4 types of the capital R, capital Y, capital R, small y, and small r, capital Y, and the small r, small y, each with the frequency of the 25% of the or the 1/4 of the total gametes production. When we write down the 4 types of the eggs and the pollen on the 2 sides of the Punnett square, it is very easy to derive the composition of the zygote that give rise to the F2 plants. And although there are the 16 squares, how many different types of the genotypes and the phenotypes are formed? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also the 9:3:3:1? The 5.3.2 is a chromosomal theory of the inheritance. Mendel published his work on the inheritance of the characters in 1865, but for the several reasons it remains unrecognized till the 19th century. Firstly, communication was not easy as it is now. In those days, his work could not be widely publicized. Secondly, his concept of the gene or the factors in the Mendel's world as a stable and the discrete unit that controls the expression of the traits and of the pair of the allele which did not blend with each other was not accepted by his contemporaries as an the explanation for the apparently continuous variation seen in the nature. Thirdly, the Mendel's approach of the using the mathematics to explain the biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though the Mendel's work suggested that the factors or the genes were discrete unit, he could not be provided any physical proof for the existence of the factors or the say what they were made of. In 19th century, the 3 scientists, the De Vries, Correns and the Van Scheckermark, independently rediscovered the Mendel's result on the inheritance of the characters. Also by this time, due to the advancement in the microscope that were taking place, the scientists were able to carefully observe the cell division, and this leads to the discovery of the structures in the nucleus and that appears to the double and the divides just before each cell division. These were called as the chromosomes or the color bodies as they were visualized by the staining. By 1902, the chromosome movement during the meiosis had been worked out. The Walter Sutton and the Theodore Boveri noted that the behavior of the chromosome was parallel to the behavior of the genes and used the chromosome movement to explain the Mendel's law. Recall that you have studied the behavior of the chromosome during the mitosis, equational division, and during the meiosis, that is the reductional division. The important thing to remember are the chromosome as well as the gene occurs in a plant. The 2 alleles of the gene pairs are located on the homologous sites on the homologous chromosomes. During the anaphase of the meiosis first, the 2 chromosomes, the comparison between the behavior of the chromosomes and the genes. The first is the chromosome occurs in the pair. The segregate at a time of the gamete formation and such that only one of each pair is transmitted to the gametes. Independent pair segregates independent of each other. And second is the gene. Gene is occur in pair and segregates at a gamete formation. During the anaphase of the meiosis first, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of the four different colors in the left and the right columns. In the left column, possibility first, the orange and the green is segregating together. But in the right hand column, the possibility second, the orange chromosomes is segregating with the red chromosome. The first possibility is the one long orange and the short green chromosome and the long yellow and the short red chromosome at the same pole. The one long orange and the short red chromosomes and the long yellow and the short green chromosomes at the same poles. The Sutton and the Boveri argues that the pairing and the separation of the pair of the chromosomes would lead to the segregation of the pairs of the factors they carried. The Sutton united the knowledge of the chromosomal segregations with the Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of the ideas, the experimental variations of the chromosomal theory of inheritance by the Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produces. The Morgan worked with the tiny fruit flies, the Drosophila melanogaster, which were found very suitable for such studies. They could be grown on the simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and the single mating could produce a large number of the progeny flies. Also, there was a clear differentiation of the sexes. The male and the female flies are easily distinguishable. Also, it has many types of the hereditary variations, and that can be seen with a low power microscope. The 5.3.3 is a linkage and the recombinations. The Morgan carried out the several dihybrid crosses in the Drosophila to study the genes, and that where the sex link, the crosses were similar to the dihybrid crosses carried out by the Mendel in peas. For example, the Morgan the hybridized yellow bodies, the white-eyed female to brown bodies, and the red-eyed males, and intercross their F1 progenies. And he observed that the two genes did not segregate independently of each other, and the F2 ratio deviates very significantly from the 9:3:3:1 ratio expected when the two genes are independent. The Morgan and his group knew that the genes were located on the X chromosome, the sector, and saw quickly that when the two genes in the dihybrid crosses were situated on the same chromosomes, the proportion of the parental gene combinations were much higher than the non-parental types. The Morgan attributes this due to the physical association or a linkage of the two genes, and coined the term is a linkage to describe the physical association of the genes on the chromosomes, and the term is a recombination to describe the generation of the non-parental gene combination. The Morgan and his groups also found that even when a gene were grouped on the same chromosome, the some genes were very tightly linked and shows a very low recombination, while the other were loosely linked shows the higher recombination. For example, he found that the gene white and the yellow were very tightly linked and shown only the 1.3% recombination, while the white and the miniature wing shown the 37.2% recombination. His student, the Alfred Stotevans, used the frequency of the recombination between the genes pair on the same chromosome as a measure of the distance between the genes, and mapped their positions on the chromosome. Today, the genetic map are extensively used as the starting starting point in the sequencing of the whole genome, as was done in case of the Human Genome Sequencing project that is described later. The 5.4 is a polygenic inheritance. The Mendel studies mainly describe those traits that have the distinct alternate forms and such as the flower color, which are either the purple or the white. But if you look around, you will find that there are the many traits which are not so distinct in their occurrence and are the separate across a gradient. For example, in a human, we don't just have tall or a short people as the two distinct alternative, but a whole range of the possible heights. Such traits are generally controlled by the 3 or the more genes and are thus called as a polygenic traits. Besides the involvement of the multiple genes, the polygenic inheritance also takes into the account and the influence of the environment. Human skin color is another classic examples for this, and the polygenic traits and the phenotype reflects the contribution of each allele. That is, the effect of each allele is the additive. To understand this better, let us assume that 3 genes, the A, B, C, control the skin color in the human with the dominant form, that is the capital A, capital B, capital C, responsible for the dark skin color, and the recessive form is a small a, small b, small c for the light skin color. The genotype with all the dominant allele, the capital A, capital A, capital B, capital B, capital C, will have the darkest. The skin color of that within all the recessive alleles, small a, small b, small c, will have the lightest skin color. As expected, the genotype with the 3 dominant allele and the 3 recessive allele will have and the intermediate skin colors. In this manner, the number of each type of the allele in the genotype would determine the darkness or a lightness of the skin in the individual. Now the 5.5 is a pleiotropy. We have to far seen the effect of the gene on the single phenotype or a trait. There are however, the instance where seen the genes can exhibit the multiple phenotypic expressions. Such a gene is called as a pleiotropic genes, and underlying the mechanism of the pleiotropy in most cases an effect of the gene on a metabolic pathways which contributes towards the different phenotype. An example of this is the disease, the phenylketonuria, which occurs in the human. The disease is caused by the mutation in the gene that codes for the enzyme is the phenylalanine hydroxylase, the single gene mutation, and this manifests itself through the phenotypic expression, the characteristic by the mental retardation and the reduction in a hair and the skin pigmentation. The 5.6 is the sex determination. The mechanism of the sex determination has always been puzzled before the genetics. The initial clue about the genetic or a chromosomal mechanism of the sex determination can be traced back to some of the experiments carried out in insect. In fact, the cytological observations made in the number of the insect leads to the development of the concept of the genetic chromosomal basis of the sex determination. The Henking in 1891 could trace a specific nuclear structures all through the spermatogenesis in the few insects, and it was also observed by him that the 50% of the sperm receives this structure after the spermatogenesis, whereas the other 50% sperm did not receive it. The Henking gives a name to his structure as the X body, but could not explain its significance. Further, the investigation by the other scientists led to the conclusion that the X body of the Henking was in fact the chromosomes, and that is why it was given the name is X chromosome. It was also observed that in a large number of the insects, the mechanism of the sex determination is of the XO type, that is, all the X bear and the additional X chromosome beside the other chromosomes, that is the autosomes. On the other hand, the sum of the sperm bears the X chromosome, whereas the sum do not. The egg fertilized by the sperm having the X chromosome become the female, and those of the fertilized by the sperm that do not have the X chromosome becomes the male. Do you think the number of the chromosomes in the male and the female are equal? Due to the involvement of the X chromosome in the determination of the sex, it was designed to be the sex chromosome, and the rest of the chromosomes were named as the autosomes. The grasshopper is an example of the XO type and the sex determination, in which the male have the only one X chromosome beside the autosomes, whereas the female have the pair of the X chromosome. These observations led to the investigation of the number of the species to understand the mechanism of the sex determination. In number of the other insects and the mammals, including the man, the XY type of the sex determination is seen where the both male and the female have the same number of the chromosomes. Among the males and the X chromosome is presenting, but its counterpart is distinctly smaller and called as a Y chromosome. The female, however, have the pair of the X chromosome. The both male and the female bears the same number of the autosomes. Hence, the male have the autosomes the plus XY, and while the female have the autosome plus the XX. In human being, in the Drosophila, and the male have the one X and the one Y chromosome, whereas the female have the pair of the X chromosome beside the autosomes. In the above descriptions, you have studied about the two types of the sex determining mechanism, that is the XO type and the XY type. But in both cases, the male produces the two different types of the gametes, either with or without the X chromosome, or some gametes with the X chromosome and the some with the Y chromosome. Such a type of the sex determination mechanism is designated to be the example of the male heterogametry. In some other organism, for example the bird, the different mechanism of the sex determination is observed. In this case, the total number of the chromosome is the same in both male and the female, but the two different types of the gametes in the term of the sex chromosome are produced by the female, that is the female heterogametry. In order to have the distinction with the mechanism of the sex determination described earlier, the two different sex chromosomes of the female bird has been designated to be the Z and the W chromosome. In these organisms, the female have one Z and the one W chromosome, whereas the male have the pair of the Z chromosomes beside the autosomes. The 5.6.1 is the sex determination in the human. It has already been mentioned that the sex determination mechanism in the case of the human is the XY type, and out of the 23 pairs of the chromosomes present, 22 pairs are exactly same in both of the male and the female, and these are the autosomes. The pair of the X chromosome are present in the female, whereas the presence of and the X and the Y chromosome are determinant of the male characteristic. During the spermatogenesis among the male, the two types of the gametes are produced. The 50% of the total sperm produces carries the X chromosome, and the rest of the 50% has the Y chromosome beside and the autosomes. The female, however, produces the only one type of the ovum with the X chromosome, and there is the equal probabilities or the fertilization of the ovum with the sperm carrying either the X or a Y chromosome. In case of the ovum, the fertilizes with the sperm carrying the X chromosome, the zygote develops into the female, that is the capital X, and the fertilization of the ovum with the Y chromosome carrying the sperm results into the male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy, there is always the 50% probability of either the male or the female child. It is unfortunately that our society women are the blame for the giving the birth of the birth to female children and have been ostracized and ill-treated because of these false notions. The 5.6.2 is the sex determination in the honeybee. The sex determination in the honeybee is based on the number of the sets of the chromosomes and individual recessive and offspring formed from the union of the sperms, and the egg develops as the female queen or workers, and an unfertilized egg develops as the male drone and by the means of the parthenogenesis. This means that the male have the half number of the chromosomes that of the female. The female are the diploid having the 32 chromosomes and the male are haploid, that is the having the 16 chromosomes. This is called as a haplodiploidic sex determination system and has the special characteristic features such as the male produces the sperm by the mitosis. They do not have the father and thus cannot have the sons, but have a grandfather and can have the grandson. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? Thus 5.7 is the mutation. Mutation is a phenomenon which results in the alteration of the DNA sequences and consequently the result in the change in the genotype and the phenotype of the organism. In addition to recombination, the mutation is another phenomenon that leads to the variation in the DNA. As you will learn in the chapter number 6, one DNA helix runs continuously from the one end of the other in the each chromatids in a highly supercoiled form, therefore lose the deletion or a gain or insertion or a duplication of the segments of the DNA, result in the alternation of the chromosomes, and since the gene is known to be located on the chromosomes, alteration in the chromosome results in the abnormalities or aberrations. The chromosomal aberrations are commonly observed in the cancer cells. In addition to the above, the mutation also arises due to the change in the single base pairs of the DNA. This is known as a point mutation. A classical example of such a mutation is a sickle cell anemia. Deletion and the insertion of the base pair of the DNA causes the flame-shaped mutation. The mechanism of the mutation is beyond the scope of this discussion. At this level, however, there are the many chemical and the physical factors that induces the mutation. There are the referred to as the mutagens. The UV radiations can cause the mutation in organism, and it is the mutagen. The 5.8 is the genetic disorders. The first is the 5.8.1 is a pedigree analysis. The idea that discovered are the disorder are the inherited has been the prevailing in the human society since the long, and this was based on the heritability of the certain characteristic features in the families. After the rediscovery of the Mendel's work and the practices of the analyzing the inheritance pattern of the traits in a human being began, since it is evident that the control the crosses that can be performed in the pea plant or some other organisms are not possible in case of the human being. Study of the family history about the inheritance of the particular trait provides an alternative. Such an analysis of the trait in several generations of the family is called as a pedigree analysis. In the pedigree analysis, the inheritance of the particular trait is represented in the family tree over a generation. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of the specific trait, abnormality or diseases. Some of the important standard symbols used in the pedigree analysis have been shown in the figure. As you have studied in this chapter, the each and every features of any organism is controlled by the one or the other gene located on the DNA present in the chromosomes. DNA is a carrier of the genetic information, and it is hence transmitted from the one generation to the other without any change or alteration. However, the changes or alteration do take place occasionally. Such an alteration or a change in the genetic material is referred to as the mutation. A number of disorders in the human being have been found to be associated with the inheritance of the changed or altered genes or a chromosome. The 5.8.2 is a Mendelian Disorders. Broadly, the genetic disorders may be grouped into the two categories, the Mendelian disorder and the chromosomal disorders. The Mendelian disorders are mainly determined by the alteration or the mutation in the single gene, and these disorders are transmitted to the offsprings on the same line as we have studied in the principles of the inheritance. The pattern of the inheritance of such a Mendelian disorders can be traced in the family by the pedigree analysis. The most common and the prevalent Mendelian disorders have the hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such a Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in a question is a dominant or recessive. Similarly, the trait may also be linked to the sex chromosome, as in case of the hemophilia. It is evidence that this linked recessive trait shown transmission from the carrier female to the male progeny. A representative pedigree is shown in the figure for the dominant and recessive trait. Discuss with your teachers and design the pedigree for the characters linked to both the autosomes and the sex chromosome. The color blindness. It is a sex-linked recessive disorders due to the defect in either the red and the green color cone of the eyes, resulting in the failure of the discriminate between the red and the green color. This defect is due to the mutation in the certain genes present in the X chromosomes. It occurs in about the 8% of the male and only about the 0.4% of the female. This is because of the gene that leads to the red gene color blindness are on the X chromosome. The male have only X chromosome and the female have the 2X. The son of the woman who carries the gene has a 50% chance of being the color blind, and the mother is not herself color blind because the gene is a recessive. That means that it is effective in suppressed by her matching dominant normal genes. A daughter will not normally be colorblind unless her mother is a carrier and her father is a colorblind. The hemophilia, the sex-linked recessive diseases, which shows that the transmission from the unaffected carrier female to some of the male progenies has been widely studied. In these diseases, the single protein that is a part of the cascade, the protein involved in the clotting of the blood, is affected. Due to this, in the affected individuals, the simple cut will result in non-stop bleeding. The heterozygous female, that is the carrier for the hemophilia, may transmit the diseases to the sons. The possibility of the female becoming the hemophilic is extremely rare because the mother of such a female has to be least carrier and the father should be hemophilic, unviable in the latter stage of the life. The family pedigree of the Queen Victoria shown a number of the hemophilic descendant as she was the carrier of the disease. The sickle cell anemia. This is an autosomes linked recessive trait that can be transmitted from the parents to the offspring, and when about the partner are carried for the gene or a heterozygous. The disease is controlled by the single pair of the allele, the HBA and HBS. Out of the three possible genotypes, the only homozygous individuals for the HBS show the disease phenotype. The heterozygous HBA-HBS individuals appear apparently unaffected, but they are carrier of the disease as there is a 50% probability of the transmission of the mutant gene to the progeny, thus exhibiting the sickle cell traits. The defect is caused by the substitution of the glutamic acid, that is the GLU, by the valine at the 6th position of the beta globulin chain of the hemoglobin molecule. The substitution of the amino acid in the globulin protein results due to the single base substitution at the 6th codon of the beta globin gene from the GAG and the GUG. The mutant hemoglobin molecules undergoes the polymerization under the low oxygen tension, causing the chance in the shape of the RBCs from the biconcave disc to elongate the sickle-like structures. The phenylketonuria, this inborn error of the metabolism, is also inherited as the autosomal recessive trait. The affected individual lacks the enzyme that converts the amino acid phenylalanine into the tyrosine. As a result of the phenylalanine is accumulated and converted into the phenylpyruvic acid and other derivatives. The accumulation of this in a brain results in the mental retardation, and they are also excreted through the urine because of its poor absorption by the kidney. The thalassemia. This is also the autosome linked recessive blood disease transmitted from the parent to the offspring when both partners are the unaffected carrier for the gene or a heterozygous. The defect could be due to the either mutation or deletion, which ultimately results in the reduced rate of the synthesis of the one of the globin chain, alpha and a beta chain that make up the hemoglobin, and this cause the formation of the abnormal hemoglobin molecules resulting into the anemia, which is a characteristic of the diseases. The thalassemia can be classified according to which the chain of the hemoglobin molecule is affected. In alpha thalassemia, production of the alpha globulin chain is affected, while the beta thalassemia production of the beta globulin chain is affected. Alpha thalassemia is controlled by the two closely linked genes, is the HBA1 and HBA2 on chromosome 16 of the each parents, and it is observed due to the mutation or the deletion of the one or the more 4 genes. The more genes affected, the less alpha globulin molecule get produces, while the beta thalassemia is controlled by the single gene, that is the HBB on chromosomes 11 of each parent, and occurs due to the mutation of the one or both genes. The thalassemia differ from the sickle cell anemia in that of the former is a quantitative problem of the synthesizing the too few globulin molecules, while the latter is the qualitative problems of the synthesizing and the incorrectly functioning globin. The 5.8.3 is a chromosomal disorders. The chromosomal disorder, on the other hand, are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. The failure of segregation of the chromatid during the cell division cycle results in the gain or loss of the chromosome, called as aneuploidy. For example, the Down syndrome results in the gain of the extra copy of the chromosome 21. Similarly, the Turner syndrome results due to the loss of the X chromosome in the human female. The failure of the cytokinesis after the telophase stage of the cell division results in an increase in a whole set of the chromosome in the organism, and this phenomenon is known as a polyploidy. This condition is often seen in the plants. The total number of the chromosome in normal human cell is 46, that is the 23 pairs. Out of these, the 22 pairs are the autosomes and the 1 pair of the chromosome are the sex chromosome. Sometimes, though rarely, either an addition, the copy of the chromosome may be included in the individual, or an individual may lack the one of any one pair of the chromosome. These situations are known as a trisomy or a monosomy of the chromosome, respectively. Such a situation leads to the very serious consequences in the individuals, and the Down syndromes, Turner syndrome, Clanford syndrome are the common examples of the chromosomal disorders. The Down syndrome, the cause of this genetic disorder is the presence of an additional copy of the chromosome number is a 21, that is the trisomy of 21. This disorder was first described by the Langdon Downs in 1866, and the affected individual is a short, saturated, statured with a small round head and furrowed tongue and partially open mouth. The palm is broad with a characteristic palm creases. Physical, psychomotor and the mental development get retarded. The Clanford syndromes, this genetic disorder is also caused due to the presence of the additional copy of the X chromosome, resulting into the karyotype of the 47, that is the XXY. Such an individual has overall the masculine development, however the feminine development of the breast, that is the gynecomastia, is also expressed. Such an individual are the steriles. Now the Turner syndrome, such a disorder is a cause due to the absence of the one of the X chromosome, that is the 45 will XO. Such a female are the sterile as ovaries are the rudimentary beside the other features, including the lack of other secondary sexual characters. Now the summary of the principle of the inheritance and the variation. The genetic is a branch of the biology which deals with the principle of the inheritance and its practices. The progeny resembling the parents in a morphological and the physiological features has attract the attention of the many biologists. The Mendel was the first to study this phenomenon systematically while studying the pattern of the inheritance in the pea plants of the contrasting characters. The Mendel proposed the principle of the inheritance, and which are the today referred to as the Mendel's law of inheritance. He proposed that the factors, the latter named as the gene, regulating the characters are found in a pair known as allele. He observed that the expression of the characters in the offspring follow a definite pattern in the different first generation, that is the F1, and second is the F2, and so on. Some characters are dominant over other, and the dominant characters are expressed when the factors are in a heterozygote conditions, that is the law of dominance. The recessive characters are only the expressed in the homozygous condition. The character never blend in the heterozygous condition. A recessive character that has not expressed in the heterozygous conditions may be expressed again when it comes the homozygous. Hence the character segregates while the formation of the gametes, the law of segregation. Not all the characters shown true dominant, the some character shows the incomplete and some shows the codominance. When the Mendel studied the inheritance of the two characters together, it was found that the factors independently assort the combine in all the permutations and the combinations. The law of independent assortment and the different combinations of the gametes are theoretically represented in the square tubular form known as a Punnett square. The factors, now known as a gene, on the chromosome regulating the characters called as a genotype, and the physical expressions of the characters called as a phenotype. After knowing that the genes are located on the chromosomes, the good correlation was drawn between the Mendel's law, that is the segregation and the assortment of the chromosomes during the meiosis. The Mendel's law were extended in the form of the chromosomal theory of the inheritance. Later, it was found that the Mendel's law of the inheritance assortment does not hold true for the genes that were located on the same chromosomes, and these genes were called as a linked genes. Closely located genes assorted together and distinctly located genes due to recombination assorted independently. The linkage map therefore the corresponding to the arrangement of the gene on the chromosome. Many genes were the link to the sexes also and are called as the sex-linked genes. The two sexes, male and the female, were found to have the set of the chromosomes which were common and another set which was different. The chromosome which were the different in the two sexes were the name as a sex chromosome. The remaining set was a name as autosomes. In human, the normal female has the 22 pairs of the autosomes and the pair of the sex chromosome, the capital X. A male has the 22 pair of the autosomes and a pair of the sex chromosome, that is the XY. In chicken, the sex chromosome in the male are the capital Z, and the female is a capital W. Mutation is defined as a change in the genetic material. A point mutation is a change of the single base pair in the DNA. Sickle cell anemia is a cause due to the change in the one base in the gene coding for the beta chain of the hemoglobin. Inheritable mutations can be studied by the generating the pedigree of the family. Some mutation involve the changes in a whole set chromosomes, that is the polyploidy, or a change in the subset of the chromosomes number, that is the aneuploidy. This help in understanding the mutational basis of the genetic disorders. The Down syndrome is due to the trisomy of the character chromosome, that is the 21, where there is the extra copy of the chromosome 21, and consequently the total number of the chromosome becomes the 47. In Turner syndrome, the one X chromosome is missing and the sex chromosome is as XO, and in the Clanford syndrome, the condition is capital XXY, and this can be easily studied by the analysis of the karyotype. Thank you so much.